Welcome and hello everyone. It's my great pleasure to introduce another speaker today, so Vincenzo De Paola. So Vincenzo is originally from Italy, but he obtained his PhD in molecular and cellular neurobiology in 2003 at the University of Basel in Switzerland. So he did his PhD under the supervision of Pico Caroni, and during his PhD his PhD thesis was on presynaptic terminal dynamics in the hippocampus. After that, for his postdoc, he moved to the US at Cold Spring Harbor with Karol Svoboda, and where he was using looking at plasticity of cortical connection using in vivo multiphoton microscopy. So in 2008, Vincenzo moved to MRC to first as a group leader and then as a lecturer, and then since 2014 as a reader, so that's corresponding, I believe, to a full professor in other places. So overall, Vincenzo is interested in studying the mechanism of synaptic and axonal reorganization in brain relevant to both aging and as well as neuropsychiatric disorders. So Vincenzo, we are very much looking forward to your talk. Thank you again for participating. So the stage is yours, so you can share your screen, and yeah, looking forward to it.
Thank you, Gaia. Thank you for the kind invitation and introduction. And thank you all for gathering here at this historic four-season landscaping venue. So today, I'd like to tell you about a new line of research that is making everyone in the lab very excited. I cannot convince anyone to work on anything different at the moment. So we study the regulation of synaptic connectivity in the brain in normal and disease states. And in particular, we study what we like to call the synaptic life cycle, that is the formation, regeneration, and elimination of synaptic connections. We and others over the last 20 years have used in vivo time-lapse imaging to discover that synaptic connections are actually plastic even in the adult brain. And these underlies cognitive processes such as learning and memory. So today I'd like to tell you why and how we study the synaptic life cycle, and in particular using mouse models. I'll then tell you about our discoveries in Down syndrome, and then how we studied wiring and dysfunction in the context of neurodegeneration. So you may not be aware that, because it's a relatively recent discovery, that synapses are structurally plastic even in the adult mouse brain, and this underlies cognitive processes like learning and memory. For example, both dendritic spines along the dendrite, which I'm drawn here, and axonal buttons along the axon continuously turn over. And this is a process that has been described in various systems from invertebrates to vertebrates, and including monkeys, suggesting that this is relevant also to humans. But why do we study the synaptic life cycle? Why is it important? Well, it turns out that mutations in over 200 synaptic genes are linked to more than 100 neurological and neuropsychiatric and neurodevelopmental diseases. And there's really no other set of proteins in the nervous system that is known to cause such a disease burden. Now, it's important to note that whether we end up with more synapses or less synapses, so with the increased synaptic density or less in the brain, it really depends on the tight balance between synapse formation and elimination. For example, in both neurodegeneration and schizophrenia, there is evidence for lower synaptic density. Now, this could be due either to decreased formation of synaptic connections or to an increase in synaptic elimination, or both. But it's very important to know whether it's one or the other or both, because the mechanisms that underlie synapse formation and the molecular mechanisms that underlie synapse elimination are very different. And so it's important to know which molecular mechanism do we need to manipulate and to rescue potential synaptic defects. Another example is in the context of aging, and from our recent work also in Down syndrome, even if the density of connections is unperturbed, and these are the most challenging cases to detect because we are mainly restricted to post-mortem analysis, so we wouldn't be able to tell, in fact, whether there's an increase or a decrease in synaptic turnover, because the density is the same, but it's just the dynamics of these connections which is changed. And finally, there's another possibility. There is increased synaptic density, which is known to be associated to other conditions like addiction and autism, but I'll be mainly focusing on this possibility here, the increased synaptic turnover and lower synaptic density. Now, how do we study the synaptic life cycles? These are very tiny microscopic structures. And the way we can study these, and other people have done this extensively as well in the past, is enabled really by a revolution in cell biology that started when Denk and Webb discovered the two-photon microscope and built the first two-photon microscope. And you need to remember here that this allows longitudinal imaging deep in the brain. So without going into the details, if you compare confocal with two-photon imaging, the difference in two-photon is that we excite only the focal spot, only your neuron, your favorite neuron of interest, only in this focal spot, whereas confocal excites fluorophores above and below the focal plane. And this, together with the fact we are using lower excitation energy, less scatter, less background, this really is ideal for high resolution imaging of structures deep in very scattering tissue, such as the brain. We have combined two-photon microscopy with transgenic lines that express fluorescent markers such as GFP in subsets of neurons, in this case, deep layer neurons in the mouse cortex. We've also coupled fluorescent markers with synaptically targeted domains, so we can study directly synaptic connections, but you can also think about doing similar experiment by tracking subcellular organelles, such as, for example, mitochondria, which can be followed in vivo and study this beautiful process of axonal transport in living organisms. And this is how we are accessing this circuitry of the cortex by swapping a piece of skull with a cover glass, a transparent cover glass that gives us a permanent window on the underlying circuitry. And this is a high magnification picture of the vessels in the brain, in a mouse brain, which are very important because they allow us to reposition the lens over the same neuron and indeed over the same synapses over extended periods of time. And what can we see? Well, that's shown here. This is an example of a stack. We're basically going inside and outside the brain. It's a fixed time point. What you can see is high-resolution images of axons and dendrites. These thinner processes are the axons. The thicker ones are the dendrites. And what we would typically do is to gather an image like this today, another one tomorrow, and then in a week, in a month, in a year, we've been able to revisit the very same piece of circuitry over a year to determine what is plastic and what is stable in these nervous system elements. What have we discovered? Well, for example, we have studied a mouse model of aging and found that in specific circuits, there is increased synaptic dynamics. So very the opposite of what we had expected before. And this increased synaptic dynamics was associated with long-term memory impairment, suggesting a new principle of long-term memory impairment. We also found that specific circuits in the brain after injury have different capabilities of regeneration. That is, the different neuron types have different regeneration capacities. And we also uncovered a rapid form of axon degeneration after injury, which was recently published. So overall, our work uncovers principles, new principles, and highlights really the critical role of the cell type in controlling synaptic plasticity in the adult brain. But there may be a problem here, and that is what we find in these mouse models may not be applicable to humans. In other words, to what extent what we study, what we discover in these mouse models is applicable to humans. And after all, the human brain has important differences when compared to the brain of other mammals, especially compared to small laboratory animals. And it's not just the size that differs, but if we consider the cortex, which is the largest portion of the human brain, there is a higher cortical thickness, more areas, more neurons, more synapses, and more neuron types. Another important difference is the timing of neural development. If we consider cortical neural progenitors, which in the cortex give rise to cortical pyramidal neurons, and with time also astrocytes and oligodendrocytes, this process takes about a week in mice, from embryonic day 11 to embryonic day 18, whereas it takes several months in humans, shown here, from gestational week 8 to gestational week 30. But how can we study synaptic connectivity and processes such as axon regeneration and degeneration in a human experimental system? Now, clearly, we cannot access the human brain, so we need to use reduced preparation. We need to use models. And a revolution came when human-induced pluripotent stem cells were discovered. And this was a true breakthrough because it allowed us for the first time to use human cells that you can get from any person, from any patient. You can get a biopsy, and these, for example, fibroblasts can be reprogrammed to become pluripotent stem cells. Now, these cells can be differentiated into any cell in the body, including neurons, including glial cells. And the promise of this technology, of this breakthrough, is that we can now, for the first time, study human-specific aspects of cortical development. We can model human brain diseases. And finally, one prospect is to develop cell-based therapies for neurological diseases such as Parkinson's. But today I'll be mainly focusing on the modeling aspect. Still, there is a challenge here because you want to really step up from in vitro preparations. Most of these neurons differentiated from pluripotent stem cells are studied typically in the dish, both as primary cultures or 2D cultures, sorry, or 3D cultures as in organoids. But we wanted to find a more physiological model to study this circuitry and the development of this circuitry. And we were lucky because about the time we were thinking about these problems, we came across a paper from the lab of Pierre van der Regen in Belgium, who had shown that by transplanting these human pluripotent stem cell derived neurons in the brain of adult mice, these human neurons would get synaptic connections that resembled and were very similar to the dendritic spines found in post-mortem human material. So we were inspired by this work and by the work of other pioneers in the brainstem cell transplantation field, and many of them are shown here. And we thought, wow, wouldn't it be amazing if we could get neurons from people with neurological conditions? And we started with Down syndrome, and I'll tell you in a minute why. And then after differentiation into cortical pyramidal neurons of the same identity as the ones that we have been studying in the mouse. And then after transplantation and the formation of this vascularized environment where not only the vasculature is present, but also the immune system is present, and typically these are missing in common in vitro preparations, then we will study the development of these human neurons, how they make connections between themselves and how activity emerges in these networks. And because these results have been recently published. I'd just like to give you a couple of highlights, and then I'll tell you how we are continuing this work. So why Down syndrome? So this is a developmental condition which, together with Fragile X, is the most common cause of intellectual disability. It's relatively common, although the numbers are going down because of prenatal screening. And it's caused by the fact that there are 3 copies of chromosome 21 instead of the typical 2 in most cells of the body, so not just the brain. It is associated with multiple developmental brain defects, including reduced brain volume, impaired neurogenesis, dendritic development, and synaptogenesis. And importantly, for what I'm going to tell you at the end, there is an increased risk of early onset Alzheimer's disease associated with Down syndrome, which means that most people with Down syndrome get an early onset form of Alzheimer's disease in their 40s and 50s, so early on, rather than in their 70s or 80s, like, for example, most people with Alzheimer's disease. Now, this may be because the amyloid precursor protein, APP gene, is present on chromosome 21, together with several hundreds of other genes. So it's a complex genetic condition which is challenging to model in mice. Even though mice and several mouse models exist of Down syndrome, taught us a great deal about Down syndrome. Most of everything that we know about Down syndrome really comes from these mouse models. But there are limitations, and one of them is that there is no chromosome 21 in rodents. And so mouse models are partially trisomic for distinct sets of genes. The Down syndrome phenotype then is influenced by a global transcriptome, not just the genes which are dysregulated on chromosome 21, but it's a global transcriptomic dysregulation. And this is why it's important to have the human genetic background. And so we think that this in vivo modeling system is ideal to study complex conditions like Down syndrome. Now, synapses are an obvious place to look for the underlying mechanisms of cognitive dysfunction in Down syndrome. But amazingly, no one had looked at the dynamics of synaptic connections, even in the many mouse models that existed. And what we found was rather surprising. We found that dendritic spines on trisomy of 21 neurons were more stable. This is an example of a time-lapse imaging. It's a dendrite. You see a dendritic branch here that has been followed in detail, and these are dendritic spines that disappeared, some appeared, and some were stable, the white ones. And quantification showed that both the survival fraction and turnover were affected, and in the same direction. That is that the survival fraction was higher and turnover rate was lower, meaning that connections were more stable. Some of these connections, dendritic spines, for example, number 1 and 2 here, we've been able to reconstruct at the electron microscopic level. They're shown here in a 3D rendering to show that they are both innervated by presynaptic terminals shown in green, suggesting that they make mature synaptic connections. But perhaps the most striking result came when we looked at network activity in these grafts. And this is an example of a activity that was monitored using the calcium sensor GCAM6, which is a proxy of neural activity. So we are seeing here bursts of activity which are hallmarks of developing neural networks. They've been detected in cortex, in thalamus, in the spinal cord, and they're thought to be important for the correct wiring of the brain. So this was sort of expected, that we would see this burst of activity, and this was very consistent with all the control experiment we've done. We repeated these using trisomic grafts, and this is what we saw. Basically, individual cells were active, and we knew that we could record synaptic input by electrophysiological recordings. But I hope you can appreciate the lack of synchronicity if you also look at the individual cell traces and compare them with the control. There is activity, but it's not synchronized. Now we're very interested in understanding why it's so. How come we have this reduced synchronicity, and is it related to the increased synaptic stability that we found in Down syndrome? In other words, how can we rescue the reduced oscillatory activity and synaptic turnover in Down syndrome? Well, we are hypothesizing that not all neurons are affected. And the reason why we are hypothesizing this is that Down syndrome people live a relatively independent life. They're happy people. And if all neurons would be affected in this way, we would expect even more severe cognitive symptoms. So how are we going to study this aspect? Well, the first thing we thought of doing is to build a map of human cell types in the Down syndrome fetal cortex by single nucleus RNA-seq. And we have accessed through a brain bank to several cases of fetal brain material. And so in collaboration with the Allen Institute for Brain Science, we're going to look at the global transcriptomic profile of these cells to find out whether there are subtypes which may be more affected than others. And we have already some hints. And so we're going to test with functional assays, two pathways. The first includes the OPEX and SAB2 genes, which are markers of basal radial glia and upper layer cortical neurons, respectively. And we're going to study these in the context of trisomy graft size, and I'll tell you what I mean in a second. And finally, we'd like to study the role of voltage-gated ion channels, which we found some of them being reduced in specifically in the trisomic grafts. But today, for time reasons, I'm only going to tell you about these OPEX and SAB2, beginning of a story. So it turns out that Down syndrome people have smaller brains. So they have microcephaly, which is already apparent during fetal development. And there's beautiful work from the lab of Mary Rutherford at King's College showed that you can relate, in fact, the volume of the brain in the fetus, which is being recorded by fetal MRI, with the performance in cognitive tests when these kids or children are born in the first years of their development. And there's really a good relationship between how well they perform in these cognitive tests and the volume of the brain in the fetus. Well, we can recapitulate this feature of Down syndrome in the graft. And what I'm showing you here are grafts shown in red with the human nuclei, in wild type in control cases here on the left. And you can see the striking difference in two trisomic individuals. And this was consistent across several experiments of transplantation, so very penetrant phenotype. Now, this could be due to a decrease in proliferation or an increase in cell death. And we tested both. And this is an example of how we studied proliferation by using the proliferation marker Ki67. An example of some of these cells here in green, and the quantification showing no difference. And the same was true for cell death. We couldn't find any difference in these late stages. So this is 3 months post-transplantation. So it's still possible that earlier on there would be a difference that could explain this reduction in size of the graft. But a clue came when we looked at these basal radial glia population, which you may know is important for the expansion of the human cortex. And this is an example of the OPEX staining in one of these grafts in control case. In the trisomy case, you can appreciate here the reduction in the human graft staying with human nuclei. And this is the merge, so there's a reduction in trisomy cases. And importantly, OPEX regulates the expression of another important gene, SAB2, which is a marker for upper layer cortical neurons. And we also find that in the graft, SAB2 is downregulated. This is in two different individuals, so 2 different donors for the grafts. And in both cases, there is a reduction in SAB2. This reduction was also seen in fetuses of Down syndrome. This is an example from a mid-gestational time point. So this is SADB2 staining, immunocytochemistry in a cross-section of the cortex. This is layer 1 here, the deep layers in a control case. And this is in a trisomic case. And you can see here the reduction of SADB2 quantified here. So what we're doing at the moment is to try to rescue these graft size phenotype by re-expressing SADB2 in the trisomic grafts or downregulating it in a control graft to see whether we can manipulate the size of the graft and we can rescue the activity phenotype that we detected. So in summary, I show you a new Vivo model of Down syndrome to study cellular phenotypes. A caveat, a limitation of this work is that, of course, we cannot model more organismal level phenotypes, such as, for example, craniofacial abnormalities or heart defects. And for these, mouse models are still essential. I showed you lower synchronized activity in trisomic neurons and reduced graft size in trisomy of 21, potentially mediated by OPEX SADB2 downregulation. Now we're very keen through collaborations to study other cell types which could be involved in the reduced synchronicity. For example, interneurons or astrocytes are likely candidates because they can regulate the activity of excitatory pyramidal neuron networks. And there's recent data that involves them in this cellular phenotype. And finally, I think it's worth mentioning that that we hope that this technique, that this approach could be applied now to different neuropsychiatric conditions. For example, autism and schizophrenia, perhaps to uncover common principles underlying cognitive impairment. So we're very excited about this follow-up avenue. And finally, in the last 20 minutes or so, I'd like to tell you how we're following up on this work to study neurodegenerative conditions. Essentially, we're trying to tackle this question here, this problem. How can we tackle axon and synaptic dysfunction in the early stages of neurodegenerative diseases? Now, this is an important question, it's an important problem, because it turns out that axon and synaptic dysfunction are nicely correlated with cognitive symptoms in neurodegenerative conditions, much better than, for example, some of the hallmarks of Alzheimer's disease, such as the accumulation of amyloid beta. So I put here a picture of an axon from a frontotemporal dementia patient. This is a post-mortem picture of an axon stained with tau, which is a microtubule-associated protein, which is mutated in frontotemporal dementia. And you can see these varicose structures, which are interpreted as a pathological hallmark. Now, because this is an endpoint analysis, it's a post-mortem material, we're really blind and we don't have answers to many key questions. For example, what precedes this varicose stage here, right? What are the preceding steps? And are there any of these steps reversible? Can they be reversed at any point? Do all varicose axons eventually degenerate? And finally, can damaged axons regenerate? And if not, can we stimulate the regeneration? And an interesting framework to study Alzheimer's disease has been put forward recently by Bardes-Trooper. And according to this view, there is a biochemical phase where key proteins, such as the amyloid precursor protein or tau, which I just mentioned, accumulate abnormally in the brain. And this happens over several decades. These toxic species accumulation then cause a so-called cellular phase where multiple elements of the nervous system are affected, not just neurons, but also the neurovascular unit, microglia, astroglia, and neurons themselves. So it's critical to understand this complex cellular reaction, and it's also critical that we use the right cellular and temporal resolution to study this complex cellular phase. And our idea is simple, is to use our recent approach to study new mechanisms of axon and synaptic dysfunction. And I told you that Down syndrome is associated with early onset Alzheimer's disease. And so we're going to use donors in this case where the diagnosis of dementia has been, is known. That is, whether it occurred early or late in their life. We're going to use also donors with mutations in the tau gene that causes frontotemporal dementia, and donors with 3 copies of APP that also undergo an early onset form of Alzheimer's disease. And then after transplantation, we're going to study axon and synaptic dysfunction. I'm going to tell you about an injury model where we can trigger axon degeneration. And so studying the susceptibility for axon degeneration. I'm also going to tell you about calcium signals and how these calcium signals can be used to inform technologies, stimulation technologies to enhance axon regeneration and try to repair these damaged circuits. But you may now wonder, okay, you told us that these human derived neurons that come from induced pluripotent stem cells are immature neurons that are useful to study neurodevelopment. So now how can we study processes which are hallmarks of age-related conditions that happen over several decades, right? Well, it turns out that in the grafts, some of these biochemical phase that I told you would happen normally over decades is highly accelerated, and we can find accumulation of beta amyloid in plaques early on, over several months inside these grafts. And this is an example of how these amyloid plaques look like in one of these trisomic grafts. And the arrangement is very similar to what can be detected in one of the most widely used mouse models of Alzheimer's disease, the 5X-FAD. And here I'm showing you a higher magnification of one of these plaques within the graft, which is stained with GFP. And that's another example. And this is now stacked through one of these plaques that surrounds nuclei, human nuclei. Now, I don't have time to show you that also the protein tau is misregulated. It's hyperphosphorylated in these grafts. And this is one of the other hallmarks of Alzheimer's disease. So what we're going to do now is to study both synaptic and network activity and the dynamics of these processes near and far from the plaques to see whether there is any effect early on these cellular processes. I need to give you now some background to explain how we are studying axon and synaptic loss. And this is an example from a mouse model. So these are mice that have been anesthetized, and then after the surgery, we've been using laser-mediated microlesion to induce axon degeneration. Basically, if this is an intact axon, we're going to park the laser in this green spot here, and we increase the power of the laser, and this is going to cut the axon, and the distal part, the part that is disconnected from the soma, is going to degenerate, whereas the proximal part can be monitored over time for regrowth and repair processes. Now, the advantage of these methodology is that it doesn't provoke the formation of a glial scar, and that is, that allows us to study intrinsic mechanisms of regrowth. Another advantage, of course, is that we are triggering the axon degeneration process. So we define the start of the degeneration as opposed to waiting an undefined period of time in non-injured conditions, right? How do we know when the axon is going to degenerate? So it's useful to be able to control the start of the degeneration. And so this is an example of one of these longer axons that have been cut here. And so the distal part is the part that is going to degenerate. And typically this happens with a gap. There's a gap in time after the lesion that precedes the degeneration. This is what we were expecting when we started this experiment many years ago now in the mouse brain. And what I'm showing you now is an illustration, a video of what we saw, which was quite surprising. So we cut this axon here on the left, and over time you can see this is the time. Over few hours, we started to see fragmentation and degeneration of the distal part. So this rapid onset form of Wallerian degeneration was unexpected, and we described it in a recent publication, and you're welcome to go to the BMC Biology website to check it out. But now back to the graft model. Now, because we were using immunodeficient animals to implant the graft, we were not sure whether the degeneration process would proceed at all, because these final phases are mediated by macroglial cells that remove the fragments. Well, it turns out that we can study human axon degeneration, and that's an example of a control graft, a human axon that has been cut here. This is the lesion side. The distal part over time, this is 6 hours, 10 hours, starts to degenerate. And this is very good news, because it means that we can study this process. And I'd like now to I'd like you to think about the degeneration process as an active process that affects locally the axon, as opposed to the soma, such as in apoptosis, but it's very similar conceptually. It locally disintegrates the axon, and it's a pathway, molecular pathway that is devoted for this process. And the reason why this is an intensively studied process is because even though the triggers may be different, right? What triggers degeneration after an injury is different from what triggers degeneration in a neurodegenerative condition. The final phases, that is this fragmentation and removal of the fragments, are conserved. And so the idea is that by studying axon degeneration due to injury will tell us something that can be applied to delay or slow down axon degeneration also in neurodegenerative conditions. For example, we have been studying calcium, which is an important factor for both for degeneration and for regeneration. In the context of degeneration, calcium after an injury, calcium influx after an injury activates proteases such as calpains, which disintegrate the axon cytoskeleton. And what I'm showing you here is a video taken from one of these control grafts that has been damaged by the laser in this band. So this bundle of axons has been cut by the laser, and you see this wave of calcium both proximally and distally. This is very typical. This is due to extracellular calcium influx inside the two cut ends of the axon. We repeated this experiment now in trisomic neurons, and this is what we saw. There was no increase in calcium proximally and distally. Instead, calcium accumulated in microdomains, which suggested to us that there is calcium dysregulation in trisomic neurons. Now, one way to offset some of the problems caused by axon loss would be if we could enhance the regenerative capacity of the damaged neurons. And so I'd like to spend the last minutes now to tell you about how we are thinking to enhance axon regeneration in these networks. Essentially, we would like to tackle this problem here, that is that the recovery from brain damage is incomplete and slow. And this is mainly due to the fact that there is limited regeneration of adult lesion brain circuitry. You may know that this is due to two synergistic mechanisms. The first is that there is an environment which is inhibitory for regrowth. The adult brain is inhibitory for regrowth. And the second is that there is a downregulation of intrinsic capability of regeneration. There's growth-associated genes which are downregulated in the adult. So back to our graft model, would human induced pluripotent stem cell-derived neurons regenerate at all after a lesion? And the answer is yes. We can study also human axon regeneration in these networks. This is an example of a human axon that has been cut here. This is the lesion site, and you can see that over a few hours there is new growth. Now, not all axons did this. Some axons did not regenerate, and in fact, most of them did not regenerate when the neurons were more mature. And I'm showing you here an example of one of these axons that was unable to regenerate. It's been cut here. That's the distal part that degenerated. And you can see the proximal part intact over up to 13 days. So this is quantified here. In young networks, 3-week post-transplantation, most axons can regenerate, whereas in mature networks, 3 months post-transplantation, most axons cannot regenerate. And this is very important because now it gives us the possibility of testing experimental strategies to enhance human axon regeneration. And how we're going to do that? Well, we're going to use electrical stimulation to enhance axon growth. And electrical activity is a signal for growth, right? Mediated by calcium signaling. But what patterns are we going to use? Well, we're going to first map the activity of regenerating axons and then impose this activity on the non-regenerating ones by optogenetic or other stimulation techniques. What I just explained to you is shown here in this cartoon. Basically, we're going to study the activity of regenerating neurons and compare it to the activity of non-regenerating neurons after an injury. Okay, so that's the laser lesion. Using calcium imaging, as I've shown you before, we can map the activity after, before and after a lesion, and we would expect that regenerating neurons would have a higher frequency, for example, of activity. And so we would learn an optimal activity pattern which is associated with regeneration. Then we're going to use this information to impose activity by using optogenetics. We can express rhodopsin in these neurons, and then by shining light at the very same frequency that was associated with regeneration, we hope to convert the non-regenerating neurons into regenerating ones, right? And we're going to be able to even trigger the formation of synaptic connections. And we're going to learn about what is important. What is the neural activity code for regeneration? Is it the spike pattern that matters, or is it the total number of spikes that matter? And here showing you real how this was done. Practically, this was the work of a postdoc in the lab, Shabana. So basically here, this is one of these human axons in one of these grafts that has been lesioned. And this shows what happens right after the lesion, this increase in calcium that I mentioned and showed you before. And this is the post-lesion, so that's the lesion site here, that's post-lesion, and these are the traces from 3 different regions of interests. This was for a non-regenerating axons, and Shabana repeated these experiments for regenerating human axons. That's an example here. Again, this is the distal part that degenerates, and the proximal part regenerates here over a few days, and these are the activity traces. To cut the long story short, this is a quantification. Basically, we found that there's a difference in the frequency in regenerating neurons, which are in blue here, for the mature group, the one that we really want to enhance the regeneration of, compared to the non-regenerating ones, which are shown here in red. And so one idea is to use optogenetics to now impose these 0.2 hertz frequency to see whether that can increase axon regeneration. But what I'd like to show you now in the last few minutes, in the last few slides, is how we are thinking about bringing these eventually to the clinic. And it's still challenging to express rhodopsins in people. It's not ideal. So we are looking for something that is more translatable. And what we are using is called temporal interference stimulation. What is temporal interference stimulation? Is a way of stimulating and driving activity in controlling activity in neurons due to interference between multiple electrical fields. So you can, for example, activate the hippocampus without activating the overlying regions, such as the cortex. And this was devised by Neil Grossman when he was a postdoc with at Boyden, and now Neil Grossman is a next door neighbor. And so with this help, we have set up a commercially available system, and we've been using it to drive activity in the human grafts. And that's an example of how we are triggering sparse activation patterns in one of these human grafts, labeled with human nuclei here. And in red, you see CFOS, a proxy of activity, a very rough label of neural activity. It tells us that these neurons have been activated, clearly not with precision that we can gain with calcium imaging. And in fact, that's the next step we're going to do. So this is just a lower magnification on the mouse brain with the human graft, and that's the merge here. And so we're going to use GCAM6 now to see whether temporal interference can alter network activity. And we're very excited about this development because, as I mentioned, once we find the right parameters to enhance axon regeneration, this is a technology that can be readily applied to people. In fact, it's been already used for the treatment of Alzheimer's disease. So finally, I'll show you that transplanted trisomic neurons, and I didn't have time, also tau mutant neurons survive, extend axons, and form connections. Axon fragmentation and altered calcium responses can be detected in these trisomic neurons. Human axon regeneration decreases with age, and that's important because now we can test various stimulation technologies to increase axon regeneration. And I also didn't have time to show you that the hallmarks of Alzheimer's disease, A-beta, tau tangles, and neural loss can be recapitulated with organoids that come from the very same line that we have been transplanting in the mouse, and this was work from our collaborator, Dean Nizetich. And finally, I'd like to thank the people who did the work, especially Shabana and Maria, who have pioneered the study of human axon degeneration and regeneration. Raquel Real, a talented neurologist who was behind the Down syndrome studies. Previous postdocs in the lab. This is a picture of the group. Our wonderful collaborators in Cambridge who have developed these beautiful human cortical neurons that we've been transplanting. Our collaborator, Dean, and Imperial collaborators. And finally, thank you for your attention. I'll be happy to take questions if there are any.
Thank you, Vincenzo, for this really beautiful talk. Time for discussion. So let me just give you, so everyone or anyone is able to unmute. So please either first raise your hand, then you can unmute yourself or write in the chat if you have questions. Maybe I start. I was wondering, I mean, this is a general question in a way, but related more to the first part. So in the Down syndrome model, you see these both reduction in network activity and reduction in cell numbers, or so at the end you don't know exactly what is apoptosis. You say that you see this reduced level of SATB2. And I was wondering whether there is a connection between the two of them. So whether, or what is the cause and what is the effect, right? So are the cells not developing or disappearing eventually at certain time points because the neuronal network activity is lower, or is the other way around, that the neuronal activity is lower because there are fewer cells?
Yeah, that's a very good question, and you put it quite well. You know, what is the cause and what is the effect? And I think one of the advantages of using this system is that we could test functionally some of these ideas. For example, we could reestablish SAB2, right, and see whether that rescues the cell numbers and whether in turn that rescues also the oscillatory activity. Or we could do the other way around, right? We could try to rescue the oscillatory activity by playing with these voltage-gated potassium channels that I mentioned, one of them HCN1, which is known to control burst activity, which we found downregulated in specifically in trisomic neurons. This was work from our collaborator, Rick Livesey, with RNA-seq. So if we put back now this HCN1 channel in the trisomic grafts and we reestablish the synchronized activity, would that have, in turn, a knock-down effect also on the size of the graft? And perhaps SAB2, which is known to be an activity-dependent gene, once these patterns of activity are reestablished, is also triggered, is stimulated, its expression is restored, and perhaps that has also an effect on the total number, on the differentiation of this population, which we think may be reduced or missing in this graft. But also, I'd like to add that why I think it's really important to study the real case scenario, which is what's happening in the fetal human cortex, right? The graft is an artificial system, and we need to know whether this reduction is confirmed in the fetal cortex. We have some evidence that this happens from immunocytochemistry, but I think having multiple cases and studying with an unbiased screen all the different all different genes and different cell types will give us a more complete picture of what really is happening at the cellular and molecular level in these early stages of fetal development in Down syndrome.
So I'm looking whether there is any question from the audience. I mean, still connected to this, I mean, maybe just a general thing, right? So I'm wondering, because, so actually there is one question, so let me favor that. So Nick, can you unmute yourself and ask the question? Let's see if it works.
Hi, yeah, I just have a question about, so some of the axons did regenerate, however, some of the axons didn't regenerate. I was wondering whether you looked into whether it was the kind of local interactions with cells that might be the difference. So whether it might have been microglia, and if you like labeled them, you could have seen them migrating to the axons that were regenerating, or perhaps there might be a spatial relation. So perhaps axons nearby to each other were more likely to regenerate together, that kind of thing.
Yeah, very good question. We don't know yet. We haven't studied the microenvironment yet. We are doing it as we speak. Shabana and Maria are working hard to try to understand what's happening in the microenvironment. Exactly as you say, is microglia stimulated and attracted to the lesion site as normally it would do in a mouse brain. Now, one caveat, of course, is the microglial cells do not come from the human implant. These are resident microglial cells. They come from a different lineage, so they are not derived from the cortical neural progenitors. But nevertheless, we know that there are microglial cells from the mouse in the graft, and the question is exactly, would they be attracted to the lesion site, and do they participate either in the degeneration process or in the regeneration process together with astrocytes and other non-neuronal cells? That's something that we are looking at the moment. Yeah.
So that's actually something that I was still wondering, right, about the microenvironment exactly. And again, going back to this activity, I was wondering, because one of the hypotheses in general is that cells that cannot really synchronize with the cells from the rest of the environment eventually are either eliminated or they become less prominent. And so I was wondering what here is the, so I don't know what is a log environment or what is a long range environment, what is winning. So I was wondering how much the other way around, how much the graft, the human graft, influences the mouse microenvironment. So who is winning? Who is stronger in a way?
Yeah, to this second part of the question, we don't know how the human graft influences the mouse brain activity just by looking at the behavior of the animals without doing any rigorous analysis. Just by looking at the animals in the cage, we couldn't tell the mice that had received a human implant from the mice that had not received an implant, suggesting that if there is an influence, it's not major. But of course, I could be totally wrong when we do systematic behavioral analysis of these mice. But the first part of the question, I think, is a fascinating aspect. You correctly said, if the synchronized activity, the general idea is that neurons that are synchronized, that fire together, they strengthen the connection between themselves, right? Whereas in Down syndrome, we seem to have found the opposite, right? Because there is the reduced synchronization, and yet the connections are more stable. So how does that square? And I think it's, we don't really have an answer to these, conundrum, if you want. But I think there's still a lot to learn as to what this synchronized activity is exactly doing in the, in the wiring of the network, and what are the relationship between this activity and synaptic dynamics. And I think having a system where we can manipulate activity and study the ensuing effect on the synaptic dynamics is very valuable.
And do you know how much of the basic properties of the, for instance, firing properties of the neurons are maintaining the graft compared to, surgery? There are very few examples, but surgery obtained as human samples that have been patched. And,
I'm not I'm not 100% sure about how they compare with surgical material. The electrophysiologist who did this experiment, Mark Smith, is really an expert, has been patching cells in the mouse, and he always admitted that these human neurons that he's been patching behave really differently in ways that are difficult to compare with what we know about mouse electrophysiology. For example, these HCN1 mediated currents, these IH-like currents, they look very different from the ones that have been detected in mice. But it's not surprising that these are different types of neurons. And with these experiments and what do we compare these recordings with is a key question, right? Because also surgical material is not ideal to know what the real electrophysiological patterns and biophysical properties of these neurons are. One thing is clear, that they appear to be there appears to be a spectrum of biophysical responses. These neurons are in different stages of development. That's clear. So they're not homogeneously mature or immature. It's a sort of a jungle of different kinds of maturity states.
Super. So thank you again a lot. I think it's time to say goodbye. It was really a pleasure to host you here, and hopefully again we'll, as I say with the others, see you in real life.
Thank you, Gaia.